Greetings and hello. Welcome, everyone. I see a number of people joining in our attendees tab here. We're so happy to have you with us. My name is Sarah David, and I am the Associate Director of the Immunology and Hematology Program at the Critical Path Institute. We thank you so much for joining the Transplant Therapeutics Consortium for this webinar today. We are very happy to have you with us here virtually to go over the sample size calculator for a surrogate endpoint in kidney transplant clinical trials. I'll have Haley go ahead and advance the slides, a couple slides here. So we can take a look at today's speakers from the Critical Path Institute. So we have Drs. Amanda Klein, the Executive Director of the Transplant Therapeutics Consortium, Bill Fitzsimmons, Senior Advisor to the Consortium, and Luke Kaczynski, Quantitative Medicine Scientist. And then on the next slide, I would also like to announce and welcome our panelists for today, Zhong Liu, Associate Director of the Therapeutic Review for the Division of Pharmacometrics at the Office of Clinical Pharmacology at FDA, and Ulf Meyer-Krisch, who's Chief Scientific Officer at Veloxis. We're really happy to have you here today. Before I hand things off to my colleagues for opening remarks, on the next slide, I will go over just a few housekeeping items. So please note that attendees to the webinar today will be muted. So we ask that you please use the Q&A function at the bottom of the screen to share with us any questions or comments you have. You can communicate with those of us behind the scenes via that Q&A tab. We will do our best to answer any webinar or Zoom related questions quickly in that Q&A chat function as they arise. But for any of the content pertaining to the sample size calculator or any of the work that the TTC has done, we'll be queuing those questions up and having our speakers answer them during the discussion session that we have allotted time for at the end of the webinar. So we look forward to your questions. Please keep those coming throughout the webinar today. And just finally, you may have heard the beep and see on your screen that we are recording this webinar, and the video recording along with the slides will be made available shortly after the webinar, and links will be emailed to all individuals who registered in short order. So with that, now I'll briefly review the agenda on the next slide. And let's see here. So we'll start out with opening remarks. And then after these remarks, there will be a presentation on the qualification of the iBox scoring system with FDA and EMA. Following that, we'll provide some information on the clinically meaningful differences in iBox parameters. And then after that, Luke and Amanda will go over the new graphical user interface that was created and provide a demonstration of how that graphical user interface can be used to run sample size calculations. And then from there, we'll move into a panel discussion before closing out the webinar for the day. So thank you for your patience as I went through some of those opening remarks. I will now pass things off to our first speaker, Amanda.
All right, thank you, Sarah. Thank you all for joining the webinar today. We are really grateful for the leadership and participation from the transplant community, as well as the current and former staff at the Critical Path Institute. Next slide. And here's a list of our acknowledgements. Next slide. The Transplant Therapeutics Consortium was formed in 2017 to accelerate the medical product development process for transplantation. This is accomplished by convening stakeholders from diverse settings, including industry, academia, clinical practice, and regulatory agencies in the pre-competitive space. The current unmet need is developing novel immunosuppressive therapies that improve long-term graft survival. And our current efforts in the TTC are geared towards long-term graft survival, specifically in kidney transplantation. Before I dive into our pursuit of qualification of the iBox scoring system, Ulf and Zhong, is there anything else you wanted to add? And thank you again for participating on the panel.
Yeah, I just wanted to reflect on the amazing progress this coalition has made and how heartening it is to see that this coalition held together. Again, it's a public-private partnership. There are many companies who are participating in this, societies, regulatory authorities, who are all working together and have been driving year after year the progress of this consortium. And really the foresight not only to drive towards hopefully eventually qualified surrogate endpoint, but also to think through the practical applications, which we are going to see a lot more today. And again, this had to be funded and this had to be driven forward. And for that reason, we are really particularly grateful for the partnership with the FDA in that. John?
Yes, thanks for Amanda's great introduction and thank you for all. I'm so happy to be a part of this great consortium, particularly focusing on the development of the quantitative kidney transplant drug development tool platforms. So as although we had achieved a great success in kidney transplantation regarding the short-term outcome based on the first year graft loss or rejection. However, there's still a strong unmet need for pharmaceutical therapies that can improve the long-term graft survival. And the Eibach, as we will discuss later, is a candidate surrogate endpoint of composite biomarker panel used in the first year after transplantation. So it's a very promising surrogate, but under the qualification process of the FDA drug development tool and the biomarker quantification program. So as a potential reasonably likely surrogate endpoint for the 5-year outcomes. So in clinical trial to support the drug application via the accelerated approval program. I believe the development of this composite system and the drug development tool platform will enhance the drug development strategy and facilitate the trial design. So I'm thrilled to be here and looking forward to hearing your comments. Thanks.
Oh, thank you both. All right, next slide. All right, so I'm going to dive into our pursuit of qualification of the iBox scoring system as a reasonably likely surrogate endpoint with FDA and EMA. Next slide. When we look into the unmet need in kidney transplantation, focusing on long-term graft survival, graft failure has significant negative outcomes: return to dialysis, decreased quality of life, retransplantation, and then subsequent decreased donor pool. Graft survival is the most important outcome for people living with a kidney transplant. When we look at current therapies, these have drastically improved short-term outcomes, like John had just mentioned. 1-year graft survival hovers around 91%. But the challenge for sponsors is executing registration trials of a feasible size to support superiority claims for 1 to 2 year studies. When we look at long-term graft survival, this remains suboptimal. The 5-year survival rate in primary kidney transplants in the United States hovers around 79%. So the challenge for sponsors is designing practical trials with a 5-year primary outcome. Next slide. The currently accepted endpoints are not sufficient to assess novel immunosuppressive therapies aimed at improving long-term graft survival. Moreover, given that the causes of late graft failure are multifactorial, predicting such failure accurately with a single marker may not be optimal. A critical path innovation meeting CPIM, was held in which FDA expressed agreement on the unmet need in transplantation and supported the qualification of a composite score as a reasonably likely surrogate endpoint for use in support of drug approvals through accelerated approval pathways. Next slide. In 2019, the Paris Transplant Group, together with members of the transplant community, published a seminal paper on the iBox scoring system. The iBox is a risk prediction tool that utilizes multiple clinically relevant features that have been demonstrated to be mechanistically associated with increased risk of late graft functional decline and failure. That is EGFR, proteinuria, donor-specific antibody, and kidney allograft biopsy histopathology. This really fully reflects the heterogeneity of graft failure. These measures are taken cross-sectionally at any time point post-transplantation. The iBox scoring system integrates these measures to generate individualized predictions of outcomes at 3, 5, and 7 years post-transplant. The iBox scoring system was designed to be used at the patient level to inform clinical care and management of transplant patients. Data prospectively collected from 4,000 consecutive subjects across 4 health centers in France were used to develop the iBox scoring system, with external validation performed in cohorts from the United States, the EU, and 3 independent phase 2 and phase 3 clinical trials. When we think about the iBox scoring system, it outperforms any of the single components of the iBox scoring system, and each component of the iBox scoring system has established clinical and analytical validity. Next slide. The iBox scoring system in the Lupi et al. 2019 British Medical Journal, as mentioned before, was designed to be used at the patient level to inform clinical care and management of transplant patients. In the TTC, we have adapted the iBox scoring system to be used as a clinical trial endpoint, incorporating feedback from both FDA and EMA. These optimizations include risk evaluation is fixed at 1 year post-transplant. This adaptation was based on feedback from EMA noting that time from transplant is an important prognostic factor, but is never affected by therapy. Therefore, having the risk evaluation fixed at 1 year post-transplant does not impact the output of the model. With the second bullet, follow-up time was set at a maximum of 5 years post-transplant, consistent with the long-term unmet need of graft failure. The third bullet, subjects who died, withdrew, lost their graft before the first year of transplantation have missing iBox scores because it's before that 12-month iBox evaluation. Therefore, analyses were performed imputing a 1 year iBox score for these subjects to allow the iBox scoring system to be used in de novo clinical trials as a primary endpoint. The imputed iBox score corresponds to a worst case scenario. In other words, the worst iBox score. And this is really to prevent survivor bias. And later on in the presentation, Luke will go through more about this imputed score. And then lastly, in this qualification submission, 2 models are included. One model with biopsy input called the full iBox scoring system, and the other model without biopsy input called the abbreviated iBox scoring system. Next slide. The iBox scoring system utilizes 4 established clinically relevant patient features that are routinely collected in transplant centers and clinical trials to compute an iBox score at 1 year post-transplant for the 5-year death-censored graft survival. These features include estimated glomerular filtration rate, EGFR, and proteinuria that are measures of kidney function, presence of donor-specific antibody, DSA, using a qualitative binary mean fluorescence intensity, MFI cutoff. And then lastly, with or without kidney biopsy histology data using BAMP lesion scores. Next slide. Shown on this slide is the abbreviated Eibach scoring system, so without biopsy. The Eibach scoring system is a composite score with a linear combination of parameters from the multivariable Cox proportional hazard model. The component measures, so eGFR, proteinuria, and DSA, were assessed at 12 months post-transplantation and used to compute the Eibach score. Because this is a composite biomarker, obtaining the data sets with the necessary variables to calculate an Eibach score was challenging, as expected. For example, urine protein to creatinine ratio was not the proteinuria measurement used consistently across data sets with all the necessary features. Analyses were conducted supporting the conversion from various proteinuria measurements to UPCR, urine protein to creatinine ratio, for use in the Eibach scoring system models. Another example is the DSA MFI. The assay was granted 510(k) clearance by FDA through CDRH as a qualitative test. Each tissue typing end user laboratory determines a laboratory specific cutoff for the presence of donor specific antibody and then validates this cutoff according to the package insert for the assay. The optimal MFI cutoff was 1400 based on literature precedent. Thus, we were able to align the analytical intended use of the DSA assay with the clinical utility in the model. We collected the assay information and laboratory certification documentation from the data sets to support this composite biomarker panel. We reviewed the documentation and deemed the analytical methods were robust, reliable, and fit for purpose. The 5-year death-censored graft survival probabilities are then estimated using the 12-month Eibach score, and this is the survival model. Next slide. The Eibach scoring system has been developed by estimating individual weights for each of the proposed components. So the eGFR, proteinuria, DSA, and then with or without biopsy, depending on the Eibach model, at 12 months post-transplant. This table shows the relative contribution of each factor in the model for the abbreviated Eibach scoring system. Next slide. We are working to qualify the Eibach scoring system with both FDA and EMA. We have an accepted letter of intent in the FDA Biomarker Qualification Program and submitted the qualification plan to FDA, so stage 2 of a 3-stage qualification process in April of this year. Additionally, we submitted the briefing package to EMA for a qualification opinion in February, also of this year. This is the first tool of its kind to seek regulatory qualification for use in kidney transplant clinical trials. The proposed context of use, shown here, provides a quantitative basis to support the use of the Eibach scoring system, composite biomarker panel, at 1 year post-transplant is a surrogate endpoint for the 5-year risk of death-censored allograft loss in kidney transplant recipients for use in clinical trials as an endpoint at a fixed landmark. The reasonably likely and accelerated approval refers to FDA terminology. Next slide. The predictive performance of the full and abbreviated Eibach scoring system was tested in the qualification derivation dataset. The T statistics gives the probability of randomly selected patients who experienced an event, so graft loss, had a higher Eibach score at 1 year than a patient who had not experienced graft loss. Both models have high C-statistic values, as shown here, 0.85 and 0.84 respectively, suggesting they can discriminate accurately within the construct of the proposed context of use. Next slide. This table summarizes the predictive performance across the validation datasets for both the full and abbreviated Eibach scoring system. The full and abbreviated Eibach scoring system models have good discriminatory ability also across the validation datasets. What I want to do is, on the upcoming slides and for the remainder of the slide deck, focus primarily on the abbreviated Eibach scoring system, so without biopsy. But please note that the predictive performance is similar to the full Eibach scoring system. Next slide. The predicted versus observed graph losses were not significantly different for the abbreviated Eibach scoring system, indicating good calibration in these data sets. The full and abbreviated Eibach scoring system models have reasonable predictive performance across the qualification data sets to support the proposed context of use. Next slide. The treatment effect was performed to demonstrate the Eibach scoring system can provide the evidence from clinical trials that a treatment effect on the surrogate corresponds to an effect on the clinical outcome. This was performed on 3 data sets, the 2 randomized controlled trials, Benefit and Benefit EXT, and a subset of subjects on a specific therapeutic regimen, IMTORI-based. The operating, predictive, and prognostic characteristics of the Eibach score show that the lower the Eibach score values, the better, and is associated with a higher expected death-censored allograft survival. The treatment effect on the Eibach score, which is negative and statistically significant in all 3 trials, corresponds to an effect on the death-censored endpoint, which is also negative in all 3 trials, and statistically significant in the Benefit RCT. In a prospective clinical trial using the Eibach scoring system, we will need to power to help ensure that the 5-year survival is significant, but these trials were not powered for that. Fine. So in summary, the proposed context of use provides a quantitative basis to support the use of the Eibach scoring system as a clinical trial endpoint at a fixed landmark. By qualifying 2 Eibach scoring system models with and without the biopsy endpoint, this will provide sponsors and investigators the flexibility in their clinical trial design with or without that surveillance biopsy at 1 year post-transplant, knowing that both models perform well for their predictive and prognostic performance. CPAS TTC submitted the qualification plan to FDA Biomarker Qualification Program in April of 2022. And so the plan is currently under active review by the agency. And likewise, we submitted the briefing package to EMA for a qualification opinion in February of this year. And we anticipate a decision from CHMP in July, so next month. Next slide. Okay, what I want to do is walk through how we translate the information, the output from a composite score into understanding what clinically meaningful differences in iBox parameters. Okay, next slide. The iBox scoring system as a composite score is not intuitive. However, the individual components are understood clinical parameters in kidney transplantation and can be used to model a minimal clinically important difference for an iBox score. So for example, an estimated glomerular filtration rate, so eGFR, difference of 5 to 10 mils per minute is considered an minimal clinically important difference for renal function. Next slide. This slide is intended to illustrate how the iBox changes for difference in iBox components, and in this example, EGFR. So we investigated the expected iBox score difference if EGFR values were changed and all other parameters remained constant. EGFR is a well-known marker of kidney function with a difference of 5 to 10 mils per minute as being clinically meaningful difference that has been defined and published. So a change of 1 mil per minute in EGFR resulted in a change of 0.046 in the iBox score, assuming all other parameters remain constant. So a difference of 10 mils per minute in EGFR results in an iBox score difference of 0.46. And again, a lower iBox score is better and is associated with allograft survival. Next slide. I'm going to turn it over to Luke to walk through how we take the information from a qualification pursuit as a reasonably likely surrogate endpoint in the context of a composite biomarker panel and create a sample size calculator to help sponsors, investigators with determining the appropriate sample size to use iBox scores in future clinical trials with the assumption it's qualified as a endpoint.
Thank you, Amanda, and hello, everyone. Can we go to the next slide, please? Okay, so as Amanda mentioned, she's kind of been covering like how we know the iBox is a good predictor of long-term graft survival, what a clinically meaningful difference is. But what I want to do here is really kind of talk about how we might be able to use that information to help answer trial design questions. Specifically, if someone's designing a trial around the iBox as this reasonably likely surrogate endpoint, how could we figure out what kind of sample size they would need, assuming you have some hypothetical two-arm trial with the difference in mean iBox scores between a treatment and a control group. So the goal here is basically just to develop a graphical user interface, a GUI, for sample size calculation using iBox scores. And I'll discuss kind of two ways to do that. The first is a standard parametric calculation, and then I'll also discuss an alternative non-parametric method. Next slide. Thank you. So just a few brief details on the parametric method. I don't want to go into like the statistics of it, but I just want to say the basics is that these methods assume some kind of distribution, in this case, a normal distribution, and then they use the difference in mean iBox scores between treatment and control group to figure out how big a sample size should be. The iBox itself is derived from the survival model. So one thing that's interesting here in the context of this reasonably likely surrogate endpoint, which would probably be used in something like an accelerated approval, where you'd have a shorter 1-year trial and then a long-term 5-year follow-up trial for kind of confirmation, is we can also get some information about that 5-year graft survival in each group. So what I'm going to be focusing on today is a 1-year trial powered by a difference in mean iBox scores between a treatment and control group. But I'll also say a few words about the difference in 5-year graft survival, because we can get a little bit of information from that because we're using a survival model. Next slide, please. Okay, so I just want to show some of the basics of this GUI we've been developing. So if you could click once, please. So the first step in this GUI, the way it would be used, since it's really just intended to, simulate a hypothetical 2-arm trial, you'd first have to select the version of the iBox. So Amanda mentioned earlier, we have a full version that includes biopsy information. And then for illustration purposes today, we're going to be focusing on the abbreviated, which excludes biopsy information and is a little bit simpler. So click again, please. Thank you. And the first thing you'd want to be able to put in is the actual mean difference in iBox scores. And that can be a little bit opaque as to exactly what that means clinically. So there'll be a little more on that in the next slide. So please click again. And then you'd have to look at the variance, which this calculates directly from the data we have. So click again, please. And lastly, you'd also put in all the typical inputs to any sample size calculation. So these are things like the superiority margin, which is, excuse me, the minimum clinically meaningful difference you'd expect to see between two groups. type 1 and type 2 error rate, and then your allocation ratio. So next slide, please. Okay, so in terms of calculating the Eibach score, as I said, and as Amanda has mentioned, knowing what clinically meaningful differences in Eibach scores can be a little opaque. So the way we have this set up is that when you click on the calculate button, this window on the right will pop up, and it'll let you put in kind of the means of each group for each of the input parameters. So for the abbreviated iBox, that's estimated glomerular filtration rate, EGFR, the urine protein creatinine ratio, UPCR, and the donor-specific antibodies, DSA. The full version would also have all those three and then include some extra biopsy variables below. The example I have set up here is a difference in 10 EGFR between the two groups. So you can see here the treatment group has 60 and the control group has 50 EGFR, and then everything else is the same. And that leads to a difference in Eibach scores of about 0.48. And as Amanda mentioned, the low Eibach score is better, high EGFR is better. So the group with the higher EGFR has the lower Eibach score, and that indicates a better chance of long-term graft survival. Okay, and next slide, please. And then once the mean Eibach difference has been decided upon, the variance can be adjusted. And as I mentioned, the variance is calculated directly from the data. So this isn't something the user would need to guess at. We actually have a data set where we can estimate the variance for the user based on whatever patient characteristics they want to exclude. So in clinical trial setting, a sponsor might want to, for example, exclude donors that are greater than or equal to 60 years old, exclude deceased donors, or exclude patients who had already had a previous kidney transplant, etc. And once those boxes are clicked on, the variance will be automatically adjusted. And as Amanda also mentioned previously, since this is a one-year trial that relies on a one-year evaluation, if there are patients who don't make it to 1 year, they can't get that evaluation. And that evaluation is what gives them their Eibach score. So if they don't get that one-year evaluation, they don't really have an Eibach score. So what we do is we give them an Eibach score that indicates very poor performance because they didn't even make it to that one-year time point. For the abbreviated Eibach, that corresponds to a worst case scenario where we have an EGFR of 0, a UPCR that corresponds to the max dipstick value, and positive donor-specific antibodies. And that gives it an Eibach score of 1.48. And then you can put in the percent estimates of like the percent of patients who have the graft loss before 1 year, percent of patients who have a death before 1 year or lost to follow-up. And then once all that's put in, that variance is automatically adjusted, and you can hit set and it will set the variance. Next slide, please. Okay, so once the Eibach score is the mean difference is set, the variance is set, and all those other parameters, so that superiority margin, type 1 error, type 2 error, allocation ratio, once all those are set, the user would hit run, and then this table would be populated. So what I have here are 4 example runs. The first one is with the full iBox. The second is the abbreviated with a 2-to-1 allocation ratio. The third is the same as the second, except now I've switched it to a 1-to-1 allocation ratio. And the last one is the same, except now that the type 2 error rate is switched from 0.2 to 0.1. And you can see this has some effects on this estimated sample size on the right. So if you could click once, please. So once this table is populated and a user has kind of filled up all the scenarios they're interested in, they can click on different rows and delete them using that red button, or they can save the whole output as a CSV. Please click again. One thing I want to highlight, though, as I've mentioned before, this is really focused on that 1-year mean difference in Eibach scores. It's not the survival at 5 years. So this sample size that it gives you is powered by this mean difference in Eibach scores. And that would be for the 1-year trial. It's not the same sample size that you'd need for that 5-year follow-up. So if you could click again, please. So the information from the iBox as a survival model can allow us to get estimates of what the 5-year graft survival will be. And these might be a little bit of a, you notice there's not a big difference between the groups here. So one thing I want to note is that to see a difference in 5-year survival, one would probably need a larger sample size than what you'd need for the 1-year difference in Eibach scores. So it would probably require, drawing from historical controls or some other way to include more patients. So I just wanted to note that, but in the context of a reasonably likely surrogate endpoint, this calculator is really focused on that 1-year trial. And then we also want to give a little extra information that might be helpful for planning a 5-year trial for that long-term follow-up. Okay, so click again, please. Okay, so that's the basics on the parametric approach, which, as I said before, it makes some assumptions about a distribution. But one thing that's really notable about the parametric approach is that when we have those imputed values, we've got those patients who don't make it to the 1 year, and we have to give them this really poor Eibach score that indicates poor performance. What we're basically doing is introducing outliers into the data. And what that does is it increases the variance. So an alternative approach is a non-parametric approach, which doesn't make the same assumptions. It doesn't assume a distribution. It relies on ranks. So it doesn't care so much that value is an outlier. It just will note that that's the lowest rank, for example. And the end result is that it's an approach that's much less sensitive to imputed values. It might even be more appropriate in the presence of imputed values. And what I want to kind of highlight on this slide is that the non-parametric approach is still a very good test of whether two groups are different. So what I have here is are the two randomized control trials, Benefit and Benefit Extend, where you can see the treatment group and control group mean Eibach scores, the mean difference in Eibach scores, and the variance. And then these last two columns are two different tests. One is a parametric test, which is a 2-sample t-test, and it finds a significant difference between the control and treatment group for each of these randomized control trials. And then I also have a Wilcox and Mann-Whitney test, which is a non-parametric, and it also finds a significant difference. So it's in the presence of imputed values, we're not really losing anything by using a non-parametric test. In fact, because it's less sensitive to those outliers, it might even be the more appropriate test. So go to the next slide, please. So if we have a hypothetical trial we're designing, and I've used data from our Benefit and Benefit Extend randomized control trials, using the low intensity belatacept and cyclosporine arms of the trials, and we can kind of set up a example of what a sample size you might need to see the difference in Eibach scores for these trials. Assuming we have a 2-sided alpha of 5%, so that's our type 1 error, a 90% power, so that corresponds to a 0.1 for type 2 error, and we've got about a 1-to-1 allocation ratio with these two arms, and then we include those imputed values for early events. And we can see here is that, you can see there's similar mean difference in Eibach scores for each of the full and abbreviated versions. The variance changes a bit, but the really thing I want you to focus on here is that the parametric total sample size estimate is actually higher than the non-parametric. So in the presence of those imputed values, the non-parametric test is actually, or the non-parametric sample size calculation actually ends up with a more efficient trial because you need fewer patients to see the same effect. So next slide, please. Okay, but actually being able to calculate a non-parametric sample size is a bit tricky. So with the parametric approach, one of the reasons it's so widely used is it's really easy to apply. You can take a mean difference between groups, know the variance, and get a sample size. For a non-parametric approach, it relies more on this idea that, like, what is the probability that if you were to randomly sample a patient from your treatment group, that it would be better than that they'd be doing better than your patient from your control group. So it's a little bit, it's not as, you can't really just give it a mean difference between groups and go from there. So the approach we're taking is actually to simulate a distribution from the GUI inputs, because we know certain things about how the distribution works. We know the Eibach scores have about a normal distribution for the 1 year, and then we have information about what the percent imputed values are. And then using that simulated distribution, we can estimate the total sample size. So I'll go into a little bit of details about how that works from the user's perspective. I'm not going to go into all the background statistics, but I'll show how this works from a user perspective. So please click again. Okay, so, and I think the main take home here will be that from a user's perspective, almost nothing's going to change. It's going to be basically the same GUI, just the way things are working is going to be a little different. So for the calculate, when a user would click on that calculate button and they'd be putting in the differences in the two groups, what those would do is give us the treatment distribution mean and the control distribution mean that we'd be using to simulate for data. So click again, please. And then we can take the variance that's calculated from the data and use that as our variance for our distributions. So if you click again, and then that information allows us to simulate a very large number of patients and using those input parameters. So a treatment control distribution mean and our variance. And click again, please. And then we can take the percent imputed values and then add that on. And this lets us create a very large data set. So I have here 10 million, because if we simulate a large enough sample, we can get a very precise estimate of sample size. And click again, please. And then once we have our simulated data set, that gets used to calculate the sample size, given the type 1, type 2 error rates, and an allocation ratio. So the main take-home message here for this other method is that for a user, just be clicking on like which method you want to use, and the way things would be happening behind the scenes would change, but the GUI itself and how it's used would be basically the same. Okay, so I think that's it from my end. I'll turn it back over to Amanda for demonstration.
All right, thanks, Luke. Okay, so let's do a demonstration where we put all the hard work into action. So I'm going to share my screen. Okay, and let's see. Can you see the GUI sample size calculator on your screen?
Yes, we can. Looks good.
Okay, great. So this is the GUI sample size calculator that we're working on. After this webinar, we will be circulating the link to it so that you can test it out as a participant and then provide us feedback as well to optimize it. So, if we were to use the Eibach scoring system as a primary endpoint in a clinical trial, we're assuming a 2-arm randomized controlled trial to demonstrate superiority of the Eibach scoring system at 1 year post-transplant in the intervention arm. So here the user will select the Eibach scoring system version. So you have the full Eibach scoring system with biopsy, abbreviated without biopsy. For demonstration, let's go with abbreviated to be consistent with the presentation. The mean Eibach difference is calculated from the mean parameter value in the control and treatment groups. These Eibach parameters can be adjusted. So we click calculate. You can see here that you have the 2-arm treatment and control, and then down the rows, EGFR, UPCR, and DSA that the user can adjust. So for demonstration, if we wanted to keep UPCR and DSA the same, but we wanted to see a clinically meaningful difference in GFR between the control and treatment groups. So let's say for the treatment group, we have a higher EGFR, which is good, of 10 mils per minute. You can see as I adjust the inputs, the mean Eibach score for each arm also adjusts. We also have reset value feature, and you'll see throughout the GUI, we have components, a hover text to help guide the user as well on many of these components. Okay, so let's hit calculate. This will automatically calculate and update the mean Eibach difference between the 2 arms based on those inputs. When we look at variance, as Luke had said, is calculated from the data that supports this graphical user interface. So let's adjust the variance. We have incorporated patient characteristics that could be considered as inclusion-exclusion criteria in a randomized prospective two-arm study in a de novo kidney transplant clinical trial. So let's hit adjust, and you'll see these patient characteristics. So for demonstration, let's select a few characteristics. So let's donor age of at least 60 years old or more. Let's select cold ischemia time of at least 24 hours, previous kidney transplant, donor-specific antibody at time of transplant. And you see also as I select these features, the adjusted variance also updates. And when we look below here, subjects who die, lose their graft, or are lost to follow-up before the first year of transplantation have those missing Eibach scores. So to account for this, as we've mentioned throughout, the user can select the imputed Eibach score corresponding to the worst case scenario, so the worst Eibach score, to prevent survivor bias. So as Luke had already mentioned, for the full Eibach scoring system, the default imputed value is 2.79. For the abbreviated Eibach scoring system, it's 1.48. But once again, the user can adjust as they see fit. And really, when we think about these inputs for percent death, percent graft loss, percent loss of follow-up, these defaults reflect what is typically observed in kidney transplant clinical trials. But once again, the user has an option to adjust those percentages. Okay, and here's the reset value if need be. Let's hit set. Okay, superiority margin, type 1 error rate, type 2 error rate have defaults because what we're looking for is superiority in the treatment arm. But once again, the user can adjust those parameters. And then the allocation ratio, this is where how you want the allocation to be for your sample size calculation. So for the first round of demonstration, let's keep it a 1-to-1 ratio, and then we hit run. Okay, and you can see up to this point, all these features are described based on the parameters summarized on the left side of the screen. When we go to the right side, you can see that for the treatment arm, the percent survival at 5 years based on those inputs is around 94.5%. For the control arm at 5 years, 92.5%. And this, remember, is reflective of just the change in the 10 mil per minute difference in eGFR with everything else held constant, and then the variance with those baseline characteristics. The associated sample size for this 1-to-1 allocation for each arm is 217 with a total sample size of 434. Now, say we want to have a 2-to-1 allocation and keep all the other parameters constant. We can also hit run. I updated the 2. And now you can see what the estimated sample size is for a 2-to-1 allocation. So it goes up a little bit. Okay. And as Luke had mentioned, you can export the results as a CSV file. If you want to delete a row, you can highlight it and then hit delete. And then it will be removed. Okay. So that is the demonstration. And like I said, we're going to circulate the link after this webinar with the recording so that the participants can test it on their own and provide us some feedback. Okay, so if we can pull up the slide deck. Excellent. All right, so really in conclusion, the sample size calculator has been developed to assist with clinical trial design using the iBox scoring system as a primary endpoint. It's available for the full with biopsy and abbreviated without biopsy iBox scoring system versions. It incorporates parameters easily adjusted to help the user make more informed decisions when they are designing their clinical trial in which they would like to use the iBox scoring system as a primary endpoint, assuming qualified. And then the sample size calculator, as Luke had said before, is driven by the mean difference in 1-year iBox scores to evaluate the drug efficacy between a treatment and control group, and then also account for subjects who die, lose their graft, or are lost to follow-up before that 1-year evaluation, and thus have missing iBox scores, really for application in a phase 3 clinical trial, de novo clinical trial. And then lastly, the current sample size calculator uses the parametric method for calculating the sample size. Next slide. So the next step, what we plan to do is incorporate the non-parametric method that Luke was presenting today to help optimize sample size calculation in the presence of missing values. And it's also an opportunity to have the sponsor or user have more options in which they would like to use a sample size calculator for their clinical trial design. We'd like to incorporate ongoing feedback from the Transplant Therapeutics Consortium, incorporate feedback from the EMA regarding their regulatory decision around the iBox scoring system, and then feedback from participants of this webinar. Our plan is to present the final sample size calculator at an upcoming in-person public workshop scheduled for September 22nd of this year. So save the date. Agenda is being developed as we speak. Okay, before we dive into the panel discussion, Ulf and Zhang, I wanted to turn it over to you if there was anything else you wanted to add.
So no questions from my side. I just wanted to highlight how important it is that the team has developed a clinically relevant interface and explanations which bring the iBox closer to clinical practice. That had been a request from the transplant community and also from the regulators to put the data, the great data we have, into context. And hopefully this is going to be enabling better clinical trial design, and it is going to incentivize ultimately more drug development and transplantation, because more drug development will also mean better prediction tools as we are moving towards investigating trial level surrogacy and other things which can solidify this endpoint. We need more trials and transplantation to occur. And again, this effort really very nicely paves the way toward further drug development and transplantation. So thank you so much.
Yeah, I have no more, no additional things to add. I just want to echo Alpha for his comments. This system, the tool platform, actually integrated the current available knowledge of the our current the best database regarding the standard of care effect size, especially for variability. So it will support the trial design and expedite the drug transplantation drug development, especially to provide the prediction for long-term benefit.
All right, wonderful. So without further ado, I see some questions popping in. So let's start the panel discussion. And so the panel discussion will be made up of me, Ulf, Zhang, Bill Fitzsimmons, and Luke Kaczynski. Okay, and Bill will be acting as the moderator.
Great. Thank you, Amanda, and excellent presentations, Amanda and Luke. Very comprehensive review of the iBox and the foundations that the GUI are built upon. As Sarah indicated at the very beginning, please feel free to type in your questions into the Q&A function at the bottom of your screen. I'll be monitoring those. If you'd like someone specifically on the panel to address your question, feel free to put that in. Otherwise, I'll try to choose the best panelists to direct the questions to. So let me start with the first question to Amanda. This is a question for you, Amanda. Has donor-derived cell-free DNA been considered as a parameter in the iBox?
The iBox scoring system was based on the data from the Loop et al. 2019 British Medical Journal publication and the data that went in there to support it. So that publication and that supporting data did not have cell-free DNA as part of it, but it could be potentially collected in the future.
Right. So our data sets don't have the donor-derived cell-free DNA in them, so we can't do the analysis. But hopefully, as the biomarker work on that and other biomarkers in kidney transplant evolve over time, we can collect that data for future analyses like you've seen today. Great. Thanks. The second question, I think, Ulf, as a transplant nephrologist as well as a pharmaceutical scientist, I think this might be a good question for you to opine upon. Ulf, are there plans to utilize the iBox in clinical practice?
So as Amanda explained, the iBox, as we are developing and putting it forward through the regulatory pathway, is really geared toward a clinical trial tool, and we are trying to enable a better ability to discern the value of potential future therapies. That said, originally the iBox was really developed by the Paris Transplant Group with very much focus on clinical practice and predicting individual patients' outcomes. That is a somewhat different context of use because that the iBox at that point may be used at different times post-transplant, and variables like time from transplant may go into this too. So it is a somewhat different approach, but certainly it has partially been envisioned originally by the Paris Transplant Group as also an individual clinical predictor tool. But again, let me make it very clear, what we are developing here in this consortium is specifically a clinical trial endpoint.
Thank you. Thanks, Ulf. The next question is for Luke, I believe. So I'll ask Luke to first opine, and then others can if they'd like to add. Luke, can you please comment on how biopsy data are incorporated into the model and how that is applied during the use of the GUI? Amanda demonstrated, and we spoke mostly about the abbreviated. So if you could go into the biopsy incorporation into the model and the application in the GUI.
Right. So in the model itself, you can think of each of these biomarkers, so EGFR and UPCR and DSA we covered today, as like kind of predictors of that long-term graft survival. The biopsy information are also predictors of long-term graft survival. So you have a number of biopsy variables, for example, IFTA, that are additional predictors in the full iBox. And so for the GUI, when you go to the calculate button, you'd see kind of the same thing. You'd see that EGFR, UPCR, and DSA, but then you'd also have all the other biopsy predictors. So IFTA would be in there, transplant glomerulopathy would be in there, just as examples. And you could set the percent of patients who are positive for those biopsy markers in each group. And then that would be factored into the full iBox sample size calculation.
Great. Amanda, is it easy to show quickly what, I believe we have enough time to do that, to show what Luke was just describing in your example?
Yes, that would be very simple. So let me share my screen. Okay, and once again, can you see my screen okay?
Yes.
Okay. So if we wanted to see the parameters for the full iBox scoring system, we select full with biopsy. We go to calculate to update the mean iBox difference. And here you see now the biopsy lesion scores, the BAMS lesion scores that are broken down based on the lesion scores that have been associated with long-term graft loss. And how they are broken down is based on literature precedent that supports this cutoff. We're developing a user guide, as well as there's some hover text that are associated, I believe, maybe not right here. So we have a user guide to describe these components. That way it also helps the user if they're not familiar with these biopsy lesion scores. Luke, anything you wanted to add to that? I think I might have cut you off.
Well, I was gonna say is I think the hover text is if you hover over the actual box where you'd input things. Yeah.
Perfect. Thank you. Okay. So there's hover text for those as well. Okay, I will stop sharing.
Okay, thank you, Amanda and Luke. The next question, I'll ask Zhang to initially respond to this one, since it includes an FDA question. Zhang, does FDA accept amending an ongoing study to include iBox scores as an endpoint?
Yeah, that's a great question. Thank you. But I must first claim that please note that the view I presented here are my personal opinion and should not be interpreted as the position of the FDA. So we have seen several submissions has proposed to use iBox score as a clinical endpoint. Either someone's proposed to use surrogate like the reasonably likely surrogate endpoint, or as a secondary endpoint, efficacy endpoint. And so far, from regulatory perspective, this is a candidate surrogate. It's not verified, validated, or so far is under the qualification process for the reasonably likely surrogate endpoints, but still it's just at the beginning of that process. So this moment, I think based on our experience, is still as a candidate surrogate. So it has been used, accepted as a secondary endpoint, but it cannot be, have not been accepted as the primary endpoint or the primary endpoint even for accelerated approval, because we still see from like an assay perspective or clinical perspective or even for statistical perspective, regulatory have multiple concerns. So we need, those concerns need to be addressed before it officially be qualified. But from my understanding, even as a secondary endpoint, it has benefit. So even the drug development tool calculated based on for the sample size based on the second point, that still can provide those kind of hypothesis generation or proof of concept to go for future like confirmatory studies.
I think the biggest part, thank you for that, Zhang, is that with the and just to piggyback, with the biomarker qualification program, it's a 3-stage process. So along the way, we receive feedback from the qualification program to optimize our submission. And so currently, like I said before, we have an accepted letter of intent, and we just submitted the qualification plan. So stage 2 of the 3-stage process, that's an active review by FDA. And then they will provide feedback that we can use to optimize the final stage of the full qualification package. So in the context of amendment, it's in the context of a qualified endpoint that we're pursuing through the biomarker qualification program, and we receive feedback along the way through these specified stages with the biomarker qualification program.
Okay, so thank you. So we could, in theory, amend a protocol now, but only as a secondary endpoint for use in primary endpoints. We would await the full qualification through the FDA. Great. The next question I'm going to address to Amanda. Amanda, can the conversions for urine protein to UPCR be shared? Will the public have access to those conversions that were used in the iBox validation work?
Absolutely. One of the wonderful parts of having a pre-competitive public-private partnership is that the work that we're doing in the TTC, if it becomes qualified, is publicly available. That doesn't mean that the patient-level data itself becomes publicly available, but the tool that, if regulatory endorsed, becomes publicly available. In the submission to FDA and EMA, this will include, and is publicly available, the documents that we submitted. That doesn't include that patient-level data, but it does include methodologies that we use to support the conversions, like the proteinuria conversion. So, if there's specific questions that someone would like and needs help navigating where to find that information, please feel free to send me an email. On our last slide, we have contact information, and happy to work through that with you.
Great. The next question is a two-part question. I think I'm going to start with the first part, directing it to Ulf. Ulf, in practice, how can iBox be used in a clinical trial? How do you envision that? As a sponsor, how do you envision incorporating iBox into your clinical trials and your development programs?
Yeah, so this question seems to be really geared towards what in regulatory terms is called the context of use. And we have certainly spent a lot of time with the Transplant Consortium to think through the appropriate context of use. So the way we currently envision the iBox to be used is as a discerning tool for a clinically meaningful difference at a year after transplantation. But this meaningful difference would then have to be corroborated with either long-term follow-up of the same patients to show that there's a meaningful difference in hard endpoints, or with a corroborating additional trial. And that's the context of use of the accelerated approval here in the US, which requires to have what's called a reasonably likely surrogate endpoint. And that is exactly what we are trying to qualify right now through the FDA, to have a qualified, reasonably likely endpoint, which will allow us to go towards this accelerated approval program. That brings up additional complexities of the long-term follow-up, which has been seen to be difficult in many therapeutic areas. And we have specific efforts to think through how potentially data could be supplemented with real-world evidence in order to strengthen the evidence of the long-term follow-up.
Thanks, Ulf. Maybe I could ask a follow-up question related to this. Although we have shown the iBox as a point in a pivotal registration trial or phase 3 trial at 1 year, do you envision it might be utilized or could be utilized in a phase 2 trial with earlier assessments than 1 year?
Certainly, dose finding efforts in earlier trials in phase 2 could potentially be aided by the iBox, especially because we have such difficulty with the traditional endpoints to discern any differences in outcomes. Again, we have to create more experience, more data around this area. but certainly you could potentially evaluate directionally, at least, even with smaller data sets, and try to extract some meaning from the iBox.
Thank you. Amanda, I think this one will be for you, and it goes again to the question about the transparency and availability publicly of information. Are the relative contributions of each variable in the iBox publicly available? And I think that that would imply the beta coefficients, the hazard ratio, some that you'd shown on the slide. Will those be publicly available, the relative contributions of each of the variables in the iBox?
Yeah, so they were showcased on this webinar, on this public webinar, and are also described in the regulatory submissions that are publicly available. The big takeaway is that the idea is to have this qualified as an endpoint for anyone who wants to use this endpoint in their clinical trial design once it's become regulatory endorsed by the agencies. And the part that's not publicly available is patient level data.
Great, thank you. This one I'll also direct it to you, Amanda, but maybe a number of us could chime in. In order to accurately reflect the current state of the art, shouldn't the control group parameters be selected to yield a 1 in 5 year survival equal to current observed values? So in other words, when you're doing the GUI, if I interpret this correctly, rather than using the iBox differences to drive that you're assuming and the baseline that you're assuming in the control group, how would it, what if you shouldn't you use the 1 in 5 year survivals in the control group as the current state of the art?
I see. I think this actually, I might lean on you to help answer, if that would be okay.
Sure. Yeah. From our perspective, what you can do is utilize the iBox GUI that was demonstrated today to estimate that 5 year survival, adjusting the inclusion-exclusion criteria and the assumptions in your control group. I believe what you should do when you're doing that is double check to see if those assumptions in terms of your trial population result in the 5 year survival that you would anticipate in the current state of the art. I think the difficulty really the difficulty in choosing the 1 in 5 year survivals and state of the art is it depends highly, obviously, upon the study population that you include, and study populations tend to do much better than the general population of transplant recipients. They're all comers. So the iBox GUI allows you to really do a better job, I think, estimating that by refining your study population based on inclusion-exclusion criteria and lots of data that predict 5 year survival. So I think it's a way that you should, I believe, use the GUI for the iBox, because that's really what you're statistically testing, that difference at 1 year. So I think that's the right approach from this clinical trial methodology. But it should be, you should verify and validate that your assumptions going into that trial are consistent with what the GUI is predicting for your 5 year survival. But Luke, anything that you would add to that?
Yeah, I just want to add one thing, which is that we have some clinical trial data where we can actually see what kind of the control group looks like in terms of the different parameters that go into the iBox. And the way we've set up the GUI is that the defaults actually reflect what we've seen in those clinical trials. So I think the defaults we put in are actually reasonable defaults for what you might see in a clinical trial setting. So there has been some attempt to do something like that. Although, as Bill said, depends on the population and other factors. So not necessarily, but we've made some attempt to make it look like what you might see in a clinical trial population.
And Luke, just to expand on this a little bit, I think the person who was asking the question was getting also at the fact that there may be an evolving standard of care, and obviously what we are observing in the GUI is really based on the data we have, which is quite dated data because we had to pick data which had at least 5-year follow-up. And by nature, this is quite old data. Now, to ease people's minds a little bit, overall in transplantation, short-term outcomes have been excellent, and they have been excellent for quite some time. Unfortunately, long-term outcomes have been dramatically lagging behind, and that hasn't changed dramatically either. And with some of the work we did in the real-world evidence group, we even found that the long-held belief that there may be dramatic differences in renal function and potentially outcomes between cyclosporine and tacrolimus that had never been borne out in prospective studies, and really in real-world data that is not borne out either. So the unfortunate news seems to be that really there haven't been that dramatic changes, which in return helps us with using this GUI that, even though it is based on historic data, it seems to be still really relevant.
Great. Thank you.
I think to summarize that is this GUI reflects the data that has all the necessary features to support a regulatory endorsed tool with baseline variables, but also parameters that go into the iBox model, and then have that 5-year follow-up reflective of a broad patient population, so living and deceased donors on different therapeutic agents that span across it's about a 20-year period for the data. So the data that's included and supports this GUI reflect the dynamics of the kidney transplant population, both transplant centers and clinical trials across the globe.
Thanks everyone for that response. Amanda, I have a question for you. We referenced the RWE work group a number of times, and I know that when Luke went through the sample size calculator and the GUI, the discussion about needing larger numbers in terms of the treatment and control arms for the survival analysis long-term was mentioned. Can you give any further insight on how you envision being able to statistically test for improvements in longer-term survival, and what the RWE work group within the TTC is pursuing?
All right, I'd be happy to. Knowing that this is something that we're actively addressing and thinking through the regulatory and design considerations for the sample size for that 5-year, the TTC established a real-world evidence work group over a year ago, in which we are evaluating the potential of supplementing internal control arms with external controls to understand the impact of new therapies on long-term graft survival. We're exploring whether registry data is appropriate for use in supplementing control arms for the long-term follow-up in kidney transplant clinical trials, assuming the iBox scoring system is qualified as a reasonably likely surrogate endpoint. So this work is underway in the TTC. We have a pilot study that we are working on the phase 2, part 2 of with UNOS. It's a collaborative effort in which we are retrospectively simulating the use of a contemporaneous external control group. So the idea of, current times, current population, as a supplement to the cyclosporine arm of the benefit RCT.
Okay. And I think, Amanda, thank you. That relates to the question that just came in terms of, have we assessed the number of patients required for the 5-year duration trial, given the predicted 5-year survival rates? So is it correct that those estimates of the number of patients will come out of the RWE work that's being done in using those supplemental external controls? Is that right?
I think that's the goal of ours. So we're first exploring whether registry data is a positive and important aspect that we can potentially do for external control arms. And so that, based on that, will help guide us on the sample size for the 5-year.
Okay. Luke, anything that you wanted to add in terms of the patient numbers required for the 5-year survival?
I don't think anything that I haven't already said. So I did mention we expect it to be a higher number of patients to do the 5-year survival follow-up. Exactly what that number will be would depend on how different the two groups actually are at the end. But yeah, I'm interested in seeing how that turns out from the real world evidence group.
Okay.
And Bill, just to comment on that, there's an additional dimension in the sample size needed. It is not only around creating the power to see a difference, but it is also around creating an adequate safety database for the regulatory authorities to assess the safety of the drug. And that will go into the individual discussions of individual sponsors with the regulatory agencies, but clearly would point towards needing larger numbers than would just be needed for a statistical difference in the iBox score.
A great point. Thanks, Ulf. Okay, the next question, I'll direct it first to Amanda, and Ulf, if you'd like to opine after that, that would be great. Once the iBox is hopefully qualified by the FDA, would it be possible to organize a TTC meeting with the FDA companies investing in drug development and kidney transplant and patients around the topic of accelerating innovations in kidney transplantation?
So I think that one of, and I'm going to harp back on the TTC and having a public-private partnership, because this is, instead of having to wait until a qualification, we can work through the whole process as now. So part of being in a public-private partnership with industry, academia, nonprofits, we also have an FDA liaison. That's part of the conversations that we have in this pre-competitive space. So ongoing dialogue that we are having in the consortium is there's a lot of unmet needs in transplant, even in kidney transplant, but in transplant in general. But how do we continue using this space to continue moving the field forward? And this is something that we're working on as we speak, as we continue moving the process forward for the iBox. But having this dialogue in this space is what we are doing on a day-to-day. And so highlighting the involvement of the TTC members and the FDA colleagues has been instrumental with moving things forward, even through the big regulatory steps. With regards to an organized meeting in the future, there is possibility of that, absolutely. I suspect that as we're building out our workshop and the agenda for the September 22nd public workshop, much of dialogue will still focus on where we are with qualification, but also dive into where we are with our real world evidence work group and other initiatives that we're exploring in the TTC. And so as more details come out, we'll be sharing those with the community.
Are there any more details, because it's related to another question, Amanda, that you could share on that September 22nd public workshop, or when do you anticipate being able to share those details?
I think the details are that right now we have it as a 4-hour public workshop that's going to be in DC. We're finalizing the location and the agenda. So we should have an idea of what the agenda looks like in the next couple months. But I would recommend individuals who are interested in part of this workshop, stay tuned and save that date.
Thanks. Ulf, anything you wanted to add to the question about this holding a combined meeting of various stakeholders, including transplant recipients, once the iBox hopefully is qualified by the FDA?
Yeah, I think this is actually going to be really important, first of all, for us to continue to better explain what the iBox means to the community and to patients, because as it was prefaced in this meeting, these scores in themselves are not as intuitive as a serum creatinine or proteinuria. So we will certainly have an ongoing effort to better explain and put data into context. And certainly, I very much sympathize with Amanda and with the person who asked the question that we need to build on the momentum and the incredible success we have had with this consortium. So we have been together now for 5 years. It has taken us a few years to even put this into place. So this is a really long-term effort. We have gained tremendous momentum, and we want to continue with this momentum. If we are lucky enough that we get this endpoint qualified, this will be an incredible success for the community and help bringing new therapies in. But our work is not done yet because we really need to make our therapies more patient-centric, and certainly getting towards looking at patient-reported outcomes and endpoints which are patient-relevant is going to be really important. And I'm trying to word this very carefully because I know how much time this is going to take. This was actually part of the original deliberations when this TTC came together to say, well, what do we do? Patient-reported outcomes were actually on the table. But looking at the timelines, we knew that with having some pre-existent data around the iBox would give us an incredible head start, and we could show a success and then hopefully have enough momentum to go towards patient-reported outcomes and really bring patients in and make all of this more meaningful for the patient community.
Yeah. Thank you. The next question, maybe I'll direct it to myself. The question is, will the RWE effort include immunosuppression data beyond cyclosporine to be considered as historical control in future trials? The short answer is yes. And in particular, what we're doing now with UNOS in our pilot study is utilizing TAC and MMF-based control because we know that that's the predominant immunosuppressive regimen utilized in de novo kidney transplant. We had Amanda referenced the cyclosporine control arm or the benefit study, and we're using that mainly because that's the RCT data that we have within the TTC that has both the iBox assessments and the long-term follow-up. But we realize that the cyclosporine control arm, which dates back in that study to as early as 2006, is no longer relevant for the majority of kidney transplant recipients. So we are actually most of our data in that pilot study that we will utilize for the RWE efforts are TAC MMF based external controls. Okay, I think I have a little bit of a prerogative as the moderator of the Q&A, and we have some time. So I wanted to direct a question to Zhang, if I could. And wondering if you could comment, because it's a little bit opaque, I think, to most panelists in terms of what happens with the review of a qualification package at the FDA in general. You don't, not specific to iBox, but who reviews it? How does that, how is that done? Can you provide any insights into the FDA review process, since the iBox is one example that's currently undergoing that review?
Yeah, I'm not in that currently in that the biomarker qualification program. So the submission was submitted to the drug development tool and the biomarker qualification program, and it will be needed by the clinical division. But I'm not currently directly involving in that review process yet. I think it's still at the early phase. We concurred the letter of intent, so that's reviewable and acceptable. we are moving to, like Amanda said, is the second stage, is the content of use. That's all I know at this moment. But then there's a interdisciplinary review team will formed together and to review the process, the qualification process, including leading by clinical, but also with clean farm by the statistics, and also for those and analytic AC methods as well.
Thank you. I realize that you're not in that specifically in that qualification group, but I think it's important, as you point out, that although the qualification group is doing the primary review, there's an interdisciplinary group that reaches out to people like you, as well as the transplant division, rheumatology and transplant medicine people, so that they can provide insights into the specifics of, in this example, the iBox and kidney transplant, or whatever the relevant experts in the FDA would be for a given therapeutic area. Thank you. We've exhausted our questions. Amanda, is there anything else that you would like to address in the Q&A discussion?
I think that, the goal of the TTC is to move the needle forward, and this can't be done by just one sponsor or one or FDA by himself, but having this pre-competitive collaboration is a shared risk and also shared reward. And this qualification has been a huge undertaking, but the goal is to help future clinical trials and transplants so that it helps sponsors and it helps regulatory with the confidence to use this as an endpoint in future clinical trials. So to help streamline that drug development process, and then as a surrogate, help with meeting that unmet need of long-term graft survival. So this is one effort. Like Bill said, we're working on our real-world evidence work group efforts as a offshoot, but still applicable for unmet need. And there's lots of opportunities that we're exploring of how to continue building the TTC in this pre-competitive collaboration to advance transplant field forward. So no other comments, just really wanted to highlight the TTC, highlight the panelists today. Thank you for your ongoing help, support, expertise, and please don't hesitate to reach out. Haley, if we can pull up the slide deck, I think that finishes the panel discussion. And then next slide, please. What we're going to do is circulate the presentation from today, as well as the recording, and as well as include website link to the Transplant Therapeutics Consortium for more information, and a link to the sample size calculator using iBox scores. As I mentioned before, any questions that come up, or feedback, or regarding the sample size calculator, or even that proteinuria imputation, or even interest in joining the TTC, please don't hesitate to send us an email. Our email is here. So with that, I think we can end this webinar, and thank you all for your participation.